Hello, good morning, everyone. I hope you had a good weekend. We are going to go ahead and start with lecture 5, which is on the topic of metabolism. Like I said before, it's a very complicated lesson. And so I think we're going to be learning it by taking notes instead of going through the PowerPoint slides. You can go through the PowerPoint slides yourself later on as well. Like most of the information we are going to be going through are written nicely in the PowerPoint as well. So, don't ignore the PowerPoint, but I'm just saying in general, people learn the topic a little bit better if they take notes from scratch. Like if I was in a classroom, I would be teaching this on the board, right? Instead of going through the PowerPoint slides. So please make sure you have some paper and some, highlighters or something like that. And we're gonna take this note. All right. So metabolism, right? Just to quickly recap a little bit for you. Okay, so metabolism is all the chemical reactions that are happening inside a cell. And there are two kinds of metabolism, right? We have the anabolism, which is building big molecules, and we have the catabolism, which is breaking down big molecules. So we had a diagram before where we have some food molecules, and these could be things like carbohydrates, right? It could be lipids, proteins, right? And even some nucleic acid, right? So that's what we ingested, and then we will be breaking it down into building blocks. Building blocks. Okay. And in the process, we are going to be giving off heat. Right? That's just going to be warming up our body. Our bodies is not going to be used for anything important. And at the same time, you are going to be releasing energy. Now, this energy that is being released can be captured, right, by ADP. Right? So adenosine diphosphate plus a phosphate would absorb that energy, and then it becomes ATP. Okay. So this ATP can be used for, I mean, it can go back to ADP and release the energy. I'll do it like that. Release the energy. Okay, and then this energy can be used for various things, right? So we can take the building blocks, for example, and we can use this energy, and then we can build molecules again, right? So this building path, that is anabolism, right? On the other hand, when we're breaking things down, that's catabolism. Okay, anabolic reaction, catabolic reaction. Now, this energy can also be used for other things, right? Put a star here. Can also be used for. Do you guys want to remind me what we can use it for? We can use it for chemical reactions, and there are two more things that we mentioned before.
Mechanical work, transport.
Yep. So I hear mechanical work and I hear transport work. Both of them are correct. Okay. So mechanical work are like movements, right? And transport as in active transport. Right? So example of active transport include things like bulk transport, protein pumps, right? So that's what this is. Okay. So today we're going to be focusing on a very specific type of catabolic reaction that is the preferred reaction our body uses for creating ATP. Okay? And it's not the first time we hear about it. Okay, so we're going to be learning about cellular respiration. Of course, all these notes are going to be posted online afterwards, but do try to copy it. Okay, don't just sit there and take screenshots or something like that. The whole point is for you to write it down so you would have a better retention of the things that you're learning. So cellular respiration, we can write a word equation. You have probably learned word equations in chemistry already. So literally, it's just words. So we have glucose plus oxygen. And then it's going to create carbon dioxide. And water and ATP. Okay. ATP again is adenosine triphosphate. 3 ATP. Okay. That's what that is. Now we can take the chemical equation. So what's the formula for glucose? Anybody remember?
C6H12O6.
That is correct. Thank you. C6H12O6. We're going to react it with oxygen. Oxygen is O2. Is one of the diatomic elements, right? Okay, it's going to have a double all the time. Carbon dioxide plus water and ATP. So we're going to balance the equation. Right? We start with one glucose molecule, and we're going to react it with 6 oxygen, which we need to breathe in. We will be exhaling 6 carbon dioxide molecule and 6 water molecule. And under ideal condition, we will be able to produce up to 36 ATP. Okay, so this is under ideal condition. Right? You might not get 36. Right? The most you can get is 36. Okay. Now, cellular respiration is what we call a redox reaction. Is a redox. You actually write it capital. Okay. So I can hear your question through the computer screen, right? What is redox? Redox reaction involves transfer of electron from one reactant to another. Again, I'm hoping that you guys remember your chemistry stuff. The ingredients are the reactants, right? And the things that you get in the end, those are the products, right? Reactants, products. So in a redox reaction, we will be transferring electron from one reactant to another. Okay, just gonna pull this down a little bit to give me a bit more space. So there are two words that you should know. Okay. When a molecule gain electron, okay, and it could be more than one electrons sometimes, it is reduced. Okay, so we can say like a reduction reaction. On the other hand, when a molecule lose electron or electrons, it is oxidized. Okay. Oxidation reaction. So when you have both of these things happening at the same time, because you transfer electron from one reactant to another, right? So the one that's losing the electron is going to be oxidized. The one that's gaining it, it's going to be reduced. So when both of them are happening at the same time, we say it is redox reaction. That's where the RED and the OX comes from. It's reduction and oxidation. I'm going to pause here for a second. Anybody want to ask anything for clarification? Great. So let's go back to our chemical reaction here. Which one is going to be oxidized? Which one is going to be reduced? Well, the is this the right color? Yeah, there we go. The glucose is the one that is going to be losing electron. Okay. And in the end, when you take enough electrons from it, it will become carbon dioxide. So this is oxidized. Okay, so glucose is oxidized into carbon dioxide. You remove electrons from glucose, and eventually, when you remove enough, it will become carbon dioxide. Where are the electrons going? Well, there's only one place to go, right? The oxygen, right? So in this case, the oxygen will pick up the electrons, and when it gains electron, when it pick up electron, it is reduced. So oxygen is reduced into water. Glucose is oxidized into carbon dioxide. And in the process, you release energy, and those energy will be captured to produce ATP. And there's a question in the chat box. Yes, redox reaction means both, right? It's a redox reaction because there is both oxidation reaction and reduction reaction happening at the same time, which kind of makes sense because you can't just gain electron, right? Like, where did the electron come from? It has to be coming from another molecule that's losing it, right? So, the redox reaction tend to happen in pairs. Does that answer your question? Anyone else want to ask anything before we move on? Okay, so that's what cellular respiration is. Let's take a closer look at what's going on in this thing. Now, even though we have a single reaction here, right, that starts with the reactants and it becomes the product, the actual reaction is not happening in a single step. Right? There are many steps in between. Okay, it's like baking a cake. Right? You start with the flour and the eggs and the butter, right? They don't they don't just magically become the cake, right? In the end, you gotta mix them together. You gotta put it in the oven. You gotta do a bunch of stuff in between before you get the final product, right? So what we see here is just the raw material and then the final product. But in between, there's a lot of steps that we have to go through. So that's what we're gonna be learning. Okay. So there are 4 stages, we call them. 4 stages in cellular respiration. Okay, 4 stages. First stage is what we call glycolysis. Okay, don't worry about what it means right now. Okay, like we're gonna explain later on. Just write down the names. Second one, it's pyruvate oxidation. And then we have citric acid cycle, also known as Krebs cycle. And then we have the 4th step, which is the electron transport chain, also known as the ETC for short. Okay. So briefly, glycolysis. Glyco means glucose. Does anybody remember what lysis mean? We learned that last class with the cell and everything. Lysis. What does it mean? No? That's okay. Lysis means to break. So glycolysis is to break the glucose molecule. All right. So we're going to break glucose into half, and then we're going to process it in the next several steps. And here is the key thing to remember. Okay, stage 1, 2, and 3, glycolysis, pyruvate oxidation, as well as citric acid cycle, right? These are the steps where we will be oxidizing glucose. Glucose is being oxidized. What does it mean? When something is being oxidized, that means we are removing electrons from it. Okay? And then we have high energy electrons are released. Okay? So as you oxidize glucose, all these very highly energized electrons are being released from step 1, from step 2, and from step 3. Okay? So we don't want to waste these energy. We don't want to waste these electrons. Right? So what we have to do is capture them. So high energy electrons are released and captured by energy carrier. Okay. So I want you to think of it like this. Okay. Like think of it as this is just like an analogy, I guess. I don't know if this is gonna be any good. Okay. So like this is building number 1. This is building number 2. Okay, like maybe this is I don't know, like this is your home, this is like a hotel, and then this is like another building, this is the mall or something. Okay. And people all want to go to the airport. Okay. Final destination. Okay, so we have all these people. Okay. They all want to go there, right? So what do? Well, you can call a taxi, right? Or like an Uber, I guess. So there's going to be a taxi that comes, right? The people get on, and then the taxi will transport them to the final destination. Okay, and then the people get off, and then the taxi goes back and pick up more people, right? So same thing over here. You might have more people here, right? So you need like a bigger taxi, a van or something. Right? Drive them to the airport, drop them off, and comes back. Same thing over here. Comes back. Okay, so in this analogy, I don't know if it's helping you guys, honestly. I just made that up on the spot. What do the people represent in this analogy compared to what I was trying to tell you here? Do you guys understand what I'm trying to say? What do the people represent?
Electrons, maybe?
Yes, thank you. That made me feel good. Okay, at least one person gets it. The rest of you hopefully also gets it, right? These are electrons. Okay, these are electrons, right? Where do they come from? Well, they came from the ripping of the glucose, right? So you will have electrons. You have people coming out from building number 1, from the mall. You have people coming out from, what did I say? Whatever. The school, okay? And you have people coming out from, houses. They all want to go to the airport, but they can't do it by themselves. Okay, so the shuttle will come and transport them. And in this case, the shuttles are my energy carrier, right? Energy carrier. Okay. Here, let's just write it down. Why not? Okay. Oh, thank you. All right. So let's put it in more scientific terms, right? Let me zoom back out. I always have a problem when I write notes like this. I just keep on zooming out and out, and eventually things get really, really small. Let me just try to shrink it a little bit and go back to proportion. All right, there we go. Okay, so there are 2 types of energy carrier. Right? Energy carrier. Okay. So NAD plus and FAD are energy carrier. That capture electrons from stage 1, 2, and 3. Okay. Don't worry about what they stand for. All right. They are just called that. And so, to keep things very simple, NAD plus will pick up electron, and then it will become NADH. Right? So like you might be thinking, hey, where did the H come from? Again, we're just trying to keep things very simple, right? Just know that NAD plus picks up an electron and it will become NADH. Similarly, FAD will pick up electron. And again, these electrons came from the glucose. It will become FADH2. You know, before they had Uber, you know, people have taxis, right? And how do you know if a taxi has passengers in it or not? Well, they have a little light on top, right? You probably have seen that in a movie if you haven't seen it in real life, right? If there are passengers, it lights up and say occupied, right? Okay, so that's like this. This is like an empty taxi with no passenger. Once the passenger, once the electron gets on, it's occupied. So the light lights up and you know it's NADH now, right? So same thing. Empty is FAD. Once you pick up the electron, it becomes FADH2. I just want to see if you guys are with me. Okay, let me launch a poll. This reaction here, either the first one or the second one, is this A, an oxidation reaction, or B, reduction reaction? What is it? Okay. Choose A if you think it's oxidation, choose B if you think it's reduction. You can always choose C if you're not sure. Okay, thank you. I think some people are still a little bit confused. So these are gaining electrons, right? So when you gain electron, it is reduction. Okay. So here we can say NAD plus and FAD are reduced. I hope you're using the same color code as me, into NADH and FADH2 respectively. Okay. And for those of you who are having trouble, I forgot to mention, people usually say like Leo and then Ger. It's pretty lame, but then you know they say like there's a tiger named Leo and then it says Ger. How does this help? Loss of electron is oxidation. Gaining electron is reduction. Okay, so that's how you can remember it if you're having trouble. So NAD plus and FAD are reduced into NADH and FADH2 when they pick up the electron. All right, so help me out here. Where will this molecule go? NADH and FADH2 takes the electrons to where? Where are we going to bring them? Where's the final destination?
Maybe it's gonna release energy.
Okay. Maybe. Anyone else? Right, like in our picture here, right? All the electrons hop on to the energy carrier, the NAD plus and the FAD. Where are we going to take them?
To the final destination.
Right, which is what? Where's the final destination? Look at your notes, guys. Sumana? Oh, I think you just accidentally unmute, probably. No? No one's with me?
Maybe to electron transport chain.
That's right, to the electron transport chain, guys. Okay, so let me just reiterate. Right, 1, 2, 3, right? That's what these 1, 2, 3s are for. That's where the electron comes from. And then where do we take them? We take them to number 4. That final destination, the electron transport chain. Okay? Do you guys understand? Please ask some question if you're confused. Okay, I'm gonna help you out because it's only gonna get more confusing. Unfortunately, this is the hardest lesson in the first unit. Okay, so just to recap, right? Tons of high energy electrons are being shot out, released from the glucose in the first step, second step, and the third step in glycolysis, pyruvate oxidation, and the Krebs cycle. All these electrons are coming out. We don't want to waste them. We're going to capture them with the energy carrier. We either capture it with NAD plus or FAD. Once we capture them, we're going to drive them all the way to the ETC, the electron transport chain. The electron will get off at the electron transport chain, and then you will have the free taxi again, right? You will have the NAD plus again. It will go back to the first three steps to get more electrons. Okay, so NADH and FADH2 takes the electrons to stage 4, right? The ETC. At the ETC, at the electron transport chain, NADH will now become NAD plus again, and then the electron gets off, right? Now this can go back. FADH2 becomes FAD and electron. Okay, so just to make it very clear for you, okay, these get on the ETC. They literally get on it. I'll tell you what that means later on, okay? And then help me out again. Where do these go? Where do these things go? Where will they go? Where do they go? Uh-oh. Is it really that bad? Where do they go? Goes back to stage 1, 2, and 3, right? To capture more electron, right? The carriers are just a shuttle, right? They just go back and forth between the first 3 stages and the ETC. Once they let the electron off, they will go back and pick up more. Just goes back and forth all day long, transporting electron. Okay? Any burning questions? Ask anything that will make you feel better about this this note so far. Okay. Well, let's do some polls question just to see where you are at. This is the question, and this is the poll. All right, just a couple more seconds. Choose something. Okay. During cellular respiration, which molecule is oxidized? Remember Leo, right? Loss of electron means oxidize. So which molecule are we ripping electrons from? We are taking it from glucose. It literally says in our notes that glucose is being oxidized. Okay, so reorganize the notes at the end of class, right? I know you have to write a quiz today, but like, you know, maybe tomorrow reorganize the notes so that it's nice and neat and you can refer to it when you need to. Okay, so that's the answer A. Let's do one more here. Again, this is like also right from the notes. But see if we can get the answer without looking. Right, couple more seconds. Correct answer is reduced, right? When a molecule accepts electron, when it gains electron, think of GER, right? GER, gaining electron is reduction. So the answer is C. Okay. Guess that's that. Now a couple more things to know here before we continue. Is the location of where these things happen. Oh, before that, actually, a few more things to know about the NADH and the FADH2. So here, these, as in the electrons, get on the electron transport chain, and its energy. Remember, these are high energy electron, right? And their energy will be used to make ATP. All right. I'll tell you how that happens. Maybe today, maybe next class. Depends on how far we go. All right, so next one is kind of important. I guess I'll write it in red. Important. Because we'll be relying on this piece of information for the rest of the lesson. One NADH carries enough energy in the form of electrons, right? Of course, to make 3 ATP molecules. Whereas 1 FADH2 carries enough energy to make 2 ATP molecules. Okay? Important to remember this. If you remember nothing else from what we talked about so far, remember this. Okay. Question. What is the maximum number of ATP that can be produced from 5 NADH and 2 FADH2? If you have the answer, type them in the chat box. Okay? But don't press enter yet. Just key it in the chat box, and then we'll press enter at the same time. So, you know, we won't see other people's answer when we're trying to figure it out. I'm just gonna give you maybe half a minute to figure this out. Type the answer in the chat box, and then you can press enter when I tell you to. Yes, 5 NADH. I know my writing is not the best. There we go. All right, if you have the answer, please press enter. Okay, great. I have a lot of people who enter something. It looks like, you guys got it right. Okay. I guess some of you are still working on it, but this is what we do. Okay, look. We have 5 NADH. Each NADH can produce 3 ATP. So if I have 5, I multiply by 3, right? 3 ATP per NADH. That's going to be 15 ATP. And then we have 2 FADH2, which I would multiply by 2 ATP per FADH2, because that's what it says here. So 2 times 2 is 4. And we add them together, right? 15 plus 4 is 19 ATP. That's the answer, 19. I can explain that again if you need me to, but I need to know what your question is, like which part it doesn't make sense. Don't be shy. If you have a question, I guarantee someone else in the class has the exact same question. You can unmute and ask me, or just type in the chat box. You have to know how to do these kind of calculations for the test. We all good?
So
Sorry, yeah, Kashana, did you say something?
no, I said I think so.
Oh, okay, thank you. Anyone else? Okay, well, don't worry, we will have lots of application on this for the rest of the note. Okay, so next we're gonna figure out the location of where these things happen. Okay. Where does each stage occur? All right. So here is a cell. This is not to scale, okay? Not to scale. What is this thing that I'm drawing? I hope you know. It's going to be on your quiz today.
Mitochondria.
Yep. Thank you. That is a mitochondria. And so this is a cytoplasm. So step one, stage one, is going to happen in the cytoplasm. Glycolysis, right? So we're going to take glucose and then we're going to split it into 2 pyruvate. More on that later, but essentially that's what happens. You split the glucose molecule into 2 molecules of pyruvate. These 2 molecules of pyruvate will go into that part of the mitochondria. Does anybody remember what we call the inside of the mitochondria? You have to know that for today's quiz as well. Right, the structure of the.
Cristae or maybe inner membrane.
Okay, so the cristae are the foldings. Yeah, that's correct. But I'm looking for the inside.
Matrix.
The matrix, that's right. So this is the matrix. All right, so the 2 pyruvate will enter the matrix, and stage 2 and 3 will take place in the matrix. That's the pyruvate oxidation as well as the Krebs cycle. And finally, these foldings, right? Those foldings are called the cristae. That is where step number 4 occurs. Okay, cristae is where you will find contains the ETC, the electron transport chain. Okay. I know it's a lot of information and seems a little bit weird, abstract, but hopefully it's making sense. All right. So just to quickly recap, right? Like, what is the point of all this? Why are we taking glucose and then, taking electrons from it and then bringing the electrons to the electron transport chain? What's the purpose? The main point of doing this is so that we can harness the energy from the glucose to produce ATP, right? Up to 36 ATP per glucose. Okay, that's the point. So the next thing we should talk about is how ATPs are actually produced. Okay, how to produce ATP. I mean, you have an idea, right? Like, you have to put the phosphate onto ADP, right? Right, but like exactly how it happens. There are two methods, okay, 2 mechanisms involved.
I'm sorry, Vincent. I missed what the 2 + 3 in the matrix represents.
Oh, stage 2 and stage 3. Okay, so I can write them out again actually. So stage one here, it's same as what we had above. Right? It's the glycolysis.
Oh, okay.
Yeah, I'll write it out. It's okay. 2 is pyruvate oxidation, which, by the way, we haven't learned exactly what's going on in them, but we will. And number 4 is the ETC, right? And for those of you who weren't here from the beginning, I said the whole note will be posted online afterwards. Okay, along with the recording, so of course. Okay, so how to produce ATP? 2 mechanisms are involved in cellular respiration. Mechanisms just means like method, right? 2 mechanisms are involved in cellular respiration. Okay, so let's talk about method number 1. Like, I'm sorry that these names are really long and complicated, but I didn't make them up. Okay, first name is called substrate level phosphorylation. We can call it SLP for short. Substrate level phosphorylation. Just gonna put a star here. Okay. To phosphorylate something means to add a phosphate. So in this case, in the context of creating ATP, what are we phosphorylating? Let me repeat my question. To phosphorylate is to add a phosphate to something. So in the context of producing ATP, what are we phosphorylating? What are we?
ATP?
That's right. So Maz, very good. We are adding a phosphate onto ADP, right? When you add the phosphate onto the ADP, like here, right? ADP plus P becomes the ATP, right? So that's phosphorylation, adding the phosphate onto the ADP. That's all it means. Like it's a long word. It looks complicated, but basically just means adding a phosphate. All right. So the first way to do this is substrate level phosphorylation. What does that even mean? Okay, so I'm just going to draw you a picture to help you understand it. It involves three things. Okay, so here we have an enzyme. Okay, and we have a donor. And this donor has a phosphate that it is willing to give up. And then over here, we have an ADP. Okay, so in substrate level phosphorylation, an enzyme transfer of phosphate from a donor molecule onto ADP to produce ATP. That's all. Nothing mysterious about it. Okay. So this molecule doesn't want the phosphate anymore. It's willing to give it up. So the enzyme literally takes it, put it onto ADP, then you get ATP. Not a common method. Only happens in stage 1 and 3. For those of you who need to write down, stage 1 is glycolysis. Stage 3 is the Krebs cycle. Okay. So it's not very common. It only accounts for about 11% of all ATP production in cellular respiration. How many ATPs do we produce in cellular respiration? What's the magic number?
36.
36, that's right. So about 4 of them are produced using this method, substrate level phosphorylation. Second method is called oxidative phosphorylation. They are both phosphorylation, right? Because they both involve adding the phosphate onto ADP. The first part of the mechanism tells you how, though, right? So in substrate level phosphorylation, we get the P from a donor molecule. In oxidative phosphorylation, there is no donor. Okay. The production of ATP is driven by oxidative reaction. In other words, reaction that involves removing electrons. Okay. Specifically, electrons removed from glucose are used to produce ATP. We actually wrote it down earlier. We just didn't give it a name, right? You already know the electrons will get on the ETC, and then their energy will be used to make ATP. Right? I wrote it down for you in purple. Okay, so that way, that method of producing ATP, we call it oxidative phosphorylation. Okay, only happens in stage 4, the ETC. Okay. Accounts for majority of ATP produced. Okay. That's the background information. Just want to see if you, once again, have any question before we take a break. When we come back, we will learn about what's going on in, glycolysis, pyruvate oxidation, that kind of stuff.
So what about the stage 2?
Oh yeah, so no ATP is produced in stage 2. Is that what you're asking?
Yeah.
Yeah. No ATP is produced in stage 2. But something does happen, which we'll learn after the break. Okay. Good question. All right. Well, I know it was a lot of writing. Your hands are probably tired. Yeah, go ahead. Yeah.
They're not gonna be on the course homepage.
Say that one more time.
I said the note is gonna be on the course homepage.
I will once I post it, I will put a link in the course homepage, and then you'll be able to access it. Okay.
Okay, thank you.
Yeah, no problem. All right, take a break. We'll resume at 9:35. All right, see you guys in a bit. Okay, welcome back. Let's keep going. So now we have an understanding of what the 4 stages are and where they happen in cellular respiration, as well as the basic mechanisms that are involved behind the process, right? Namely, we're going to be removing electrons from glucose in first 3 steps. And then the energy carrier will take these electrons to the electron transport chain. Along the way, we will be making a little bit of ATP through substrate level phosphorylation. Not a lot, just about 4 of them in stage 1 and stage 3. But the bulk of the ATP, a majority of the ATPs, are actually produced by those high energy electrons that we remove from glucose, we're going to be using them to make a lot of ATP in the last step in the electron transport chain. So right now, what we're going to do is we will try to take a closer look at what's happening in each of the stages. All right, starting with the first stage. Okay, so first stage is glycolysis. Okay. So glycolysis is basically, again, two words, right? Glyco means sugar. Lysis is to break. So this is glucose. This is to break. Okay, so in glycolysis, glucose molecule is broken down into two pyruvate molecules. So glucose becomes pyruvate, and pyruvate. We already know glucose has 6 carbon atoms in it. And when you break it down into 2 pyruvate, each pyruvate molecule would have 3 carbons in them. I need you to keep track of the carbon atoms, because ultimately all these carbon atoms in glucose will end up in what other molecule? Let me repeat my question. All of the carbon atoms in glucose will ultimately end up in what molecule? That's okay. Let's go back here and take a look. In our overall equation, we can see glucose, C6H12O6, will be oxidized into carbon dioxide, right? 6 of them. So all the carbon atoms in glucose will ultimately end up in the carbon in carbon dioxide. Does that make sense? That's what I was asking you, right? All 6 carbon ultimately will end up in what molecule? They would ultimately end up in carbon dioxide. Okay. So for now, they are not in carbon dioxide yet. They are just going to be in the pyruvate. Okay. So that's the overall thing that's going to be happening in glycolysis. So a couple of other things are going to be happening. The glycolysis is broken down into 2 phases. Into 2 phases. One, the energy investing phase. And number 2, energy yielding phase. You know, you might have heard the phrase before, it takes money to make money. That certainly is true when you invest money in the bank, right? Or you buy some investment, some stock, later on you will get return, right? Hopefully you'll get return. So in this case, in glycolysis, we are investing a little bit of energy into the molecule first, and then we will end up getting some energy back. So glycolysis is actually broken down into 10 steps. The first 5 steps is are the energy investing phase. Okay, so 2 ATP molecules are used to energize glucose. I don't know if you remember, but like in lecture 2, right? I showed you this graph before. Like this is energy, right? This is time. And then the graph looks like this, right? Does anybody remember seeing this? What do we call this part of the graph? What kind of energy was that? Do you remember? No? But do you remember seeing this graph though?
Yeah, oxidation energy.
The what energy, sorry?
Oxidation energy.
Close. This is the activation energy, right?
Yeah, activation energy.
Activation energy. That's the energy that's required to kickstart a reaction, right? So that's essentially it's what this is. The energy investing phase is the equivalent of activation energy. Right? We are investing 2 ATP molecules to energize the glucose, to make it want to react. Okay, and then in the end, the energy yielding phase, what are we gonna get? We're gonna get 4 ATP molecules back. 4 ATP molecules are produced by substrate level phosphorylation, right? The last thing that we did before the break, right? We're gonna have some donors. They are gonna be giving us the phosphate, which allow us to produce 4 ATP. And that's not all, right? As far as investment goes, we got our 2 ATP back, and then we get 2 additional ones, so that's good. But in the energy yielding phase, high energy electrons are released as well. And captured by NAD+. So 2 NADH are produced. Okay. So main products of glycolysis. We have 2 pyruvate. Okay. We have 2 net ATP and 2 NADH. Okay. Remember, we actually invested 2 ATP to start the process. So even though we produce 4 in the yielding phase, overall, we are only gaining 2, right? That's why it says 2 net ATP. And you get 4 gross, 2 net. Gross is before you subtract your expenses, right? So 4 gross, 2 net.
So you put 2 in and you're getting 4.
4 back, that's right. So
So
in the end,
you
how
get
much
into
did you actually make?
2. Okay.
Yeah, exactly, right? So these are the main products. Now, at this point, which molecule has the most energy? Right? Let me give you a multiple choice question here. At the end of glycolysis, which of the following molecule has the most chemical energy? This one probably requires a little bit of thinking. Okay, I didn't get a lot of responses, but I guess that's okay. I will end the poll. Yeah, most people choose ATP, right? That's the common answer. But unfortunately, that's not the correct one. The correct answer is pyruvate. Right? Let me explain to you why. So sure, ATP has a lot of energy because we keep on saying, oh, we want to make ATP so that we can power other things, right? Okay. But remember, each NADH is capable of creating how many ATP? Do you guys remember the note we took before the break? How many ATP can you make with just one NADH?
3.
3, right? So, comparing the energy worth, the energy chemical, NADH already has more chemical energy than ATP because ATP is just one ATP, but each NADH can make 3 ATP, right? So energy-wise, NADH already has more energy than ATP. But let's take a look at pyruvate. Right? We are finished with the first step of cellular respiration. We still have step 2 and step 3 and step 4, right? And in total, we are supposed to make up to 36 ATP. We have only made 2 ATP so far. So where does the rest of the 34 ATP come from? Well, we have to continue to break down pyruvate further, right? That's where you're going to get the rest of the ATP, right? So the correct answer is pyruvate. It has the most chemical energy, right? Contains the most chemical energy at this point, right? We still haven't used up all of its energy. We still can break down pyruvate further, make more ATP, right? Make more energy carrier, right? So the correct answer is pyruvate. Does that make sense? Okay, I hope it makes sense.
Yes, sir.
All right, thank you. Always good to hear some confirmation. All right. For those of you who are still a little bit confused, I don't know. I just thought of an analogy. So it's like buying a carton of juice or something, orange juice. Okay, orange juice. And it's this full. Okay, orange juice. Right? So like you pour out a cup. Okay, you pour a cup. Right, so it goes down a little bit, right? Okay, and then you pour out like maybe you want to pack some to school or something. Right? Pour out like a bottle, right? So like that. Right. So at this point, which one has the most energy? It's still the cotton, right? The cotton still has the most juice, right? You pour out a cup, great. You pour out the bottle, right? Use up some. The bottle can maybe pour out 3 other cups, okay? But in terms of which one still has the most juice in it, is the original, right? So that's the pyruvate. You used up some energy to make these things. You still have a lot to go, right? We can break it down further. We can pour more cups later on in stage 2, stage 3, and stage 4. All right. If you think of a better way to explain this, please let me know. But that's it. That's it. And by the way, we wrote it down already, but I just wanna reiterate. Where does this happen? Where does this glycolysis take place? Takes place
Respiratory,
in the where? Sorry.
respiratory system.
In the cytoplasm.
Yeah, in the cytoplasm. That's right, in the cytoplasm. Yeah. So the respiratory system allows you to bring the oxygen into the body, which then you will use for cellular respiration. But specifically, the glycolysis, the breaking of the glucose into the pyruvate, that takes place in the cytoplasm. Okay, we had a diagram earlier, right? Just as a reference, right? Right here. Step 1 happens in cytoplasm. 2 and 3 happens in the matrix, and ETC stage 4 happens in the ETC. Happens in the quiz state. Okay, so that's that. All right, moving on. Next thing that we're gonna do is the hold on. Next step is going to be pyruvate oxidation. Did I put a 1 here? Here, I'll put a 1 here. That's glycolysis, stage 1. Stage 2. Pyruvate oxidation. Now, without me telling you anything, the name of this stage actually tells you what's going to happen. We got the pyruvate from the first step, and now we're going to oxidize it more. What does it mean to oxidize pyruvate? What are we going to do to it?
The release of electrons.
That's right. We're going to remove more electrons from it, right? So pyruvate molecules, you got 2 of them, remember, okay? Produced from glycolysis enters the mitochondrial matrix. Okay. And then there are 3 steps involved. Okay. So pyruvate. It has how many carbons again? Just think about it. Hopefully you said 3.
3.
Yep, thank you, Mona. And then we're gonna do 3 things to it. Okay. So I'm just gonna draw it out for you. First, we are going to remove a carbon dioxide. Okay, so this is called decarboxylation. Removing the carbon dioxide. Okay. Secondly, we are going to, as we said earlier, oxidize the pyruvate, which means you will release high energy electron. Every time we release high energy electron, we will need to capture it with energy carrier, right? Such as NAD+. Right? Capture it, and then it will become NADH. Okay. So at this point, we started with glucose, and then we did a lot of reactions along the way already, right? We made some ATP, we made some NADH. So by now the molecule is kind of running out of juice, so to speak, right? In our analogy, we're losing the initial energized form, right? Remember, at the very beginning, we invested 2 ATP to energize the molecule, right? But then we produce so much ATP, we rip so many electrons from it. By this point, the molecule is not really willing to react further. Okay? So what do? We want to re-energize it. We want to recharge it so that it can react more in stage 3. And the way to do it is we're gonna add something. We're gonna add something to it. And that something is called coenzyme A. Okay, so coenzyme A, the purpose of coenzyme A is to recharge the molecule so that it is going to be willing to react further. Okay. Let me just say that one more time, like maybe slightly different. in a slightly different way. We started with glucose, and in order for us to get glucose to react, we want to charge it up, right? We want to energize it. How did we do that? We invested 2 ATP, right? So the molecule is energized now, and then we did all these things in the yielding phase. We make 4 ATPs. We release high energy electron. We produce some NADH. And then it becomes the pyruvate. And then here in the second step, we take that pyruvate, we remove a carbon dioxide, we release more energy. Now the molecule is not as energized as it needs to be. So we want to recharge it, we want to re-energize it. How do we do that? We give it something called the coenzyme A. Okay, never mind what it really is. We don't have to know it. We just have to know that this molecule recharges the pyruvate. And at the end of it, you get something called acetyl-CoA. So please help me out here. Help me see that you understand what I'm talking about. How many carbons, original carbons, will we find in acetyl-CoA? We started with 6 in glucose. How many carbon atoms do we have now in acetyl-CoA?
2.
That is correct. 2 carbon. Right? Because we have 3 in the pyruvate, you remove a carbon dioxide. 3 minus 1 gives you back the 2. Okay? That's what happens in pyruvate oxidation. Now, how many times does this reaction happen for each glucose?
3 times. Maybe.
Did you say 3 times? I didn't
Yeah.
quite hear.
3 times.
Yeah. Okay. Anyone else? The question is, how many times does this reaction happen for each glucose that we break down?
2 maybe.
2. Okay. Anyone else? See, one of the drawbacks of online learning is I cannot see your face, so I cannot read your expression. I don't know if you don't get it, or like if you're just not telling me, right? But this is gonna happen 2 times, right? 2 times for each glucose. If you're not sure why, that's because the glucose, which has 6 carbon, is going to be broken down into 2 pyruvate, right? In glycolysis. Each of those pyruvate molecule will undergo the same thing. Okay, so in the end, you would have 2 acetyl-CoA. You will remove 2 carbon dioxide, and you know, you will create 2 NADH. All right. Yeah, don't worry, there are more poll questions coming up. All right. But it's better if you get, like, if there's something that bothers you guys, right? Like that you don't, it's not clear, it's better if you ask me, like, you know, give me a question. By the way, this is like my second semester was just telling me last week that, you know, this lecture was the hardest in one of the hardest in the first semester, right? And then there's also biotechnology, which is also very difficult. Those are the 2 things, right? But I want you to know this is standardized curriculum. You take pre-health at any of the Ontario college, you're gonna have to learn these things, right? So it's, you know, don't get the wrong idea. It's not that I'm forcing you to learn this. It's part of the program. Okay, so don't blame me. Blame the ministry, if anything. So that is pyruvate oxidation. Okay, and again, this happens in the matrix, right? In the mitochondrial matrix. No ATP is produced in this particular step. All right.
Professor,
Yep, go ahead.
if no ATP is produced in this reaction, we should go until we get the ATP, right?
If no
Break it
ATP is
down.
produced in this step, and then I missed the second part of your question.
So shouldn't we like break it down more to get ATP, or should we stop here? Like,
Yeah, you're right. Okay, so like we are not done with the molecule, right? So the acetyl-CoA will now proceed to stage 3, where it will be broken down further. And let's not forget, guys, what's going to happen to these NADH? Okay, what's going to happen to these NADH? Someone tell me.
We're gonna go to the final destination.
You're going to go to the final destination. Exactly. The ETC, right? The electron transport chain. And that's where the money is, right? That's where you will be making a lot of ATP, right? Using oxidative phosphorylation. So what are the key products in pyruvate oxidation? We get 2 carbon dioxide. We have 2 NADH. And we have 2 acetyl-CoA. Okay, that's the key product. All right, hang in there. I'm just gonna quickly go through stage 3, and then I will let you go. Okay, so remember, these are the main product. And if you need to remind yourself, right? Like, I'm just gonna highlight this guy. Like, where is this gonna go? Right, goes to ETC. Right. Seriously, like you got to reorganize this note in a way that makes sense to you, right? Use the proper color coding, right? Goes to ETC, final destination in the cristae of the mitochondria. All right. Now, stage 3, the last thing that we will learn today. That is called the citric acid cycle or Krebs cycle. Just gonna write Krebs cycle here. Takes place in the matrix.
Sir, could you also write slash citric acid cycle beside Krebs?
Sure. Krebs cycle, citric acid cycle, right? There we go.
Thank you.
Okay. Trish, you have something you want to add?
Yeah, I was just wondering if there's a study group for this class, like with anybody that understands the concepts well.
Oh, like with you
Sometimes
amongst the students? Is that what you mean?
Yeah.
You guys are free to set up your own study group. In fact, I can
I was
set
just
up.
asking in general. Maybe somebody could answer in the chat if I could join your group or something like that.
Yeah, you know what? I can create like a online space for you guys to post questions, and I can monitor it as well.
Yes,
And I can
yes,
answer. Yeah,
that
I'll
would be
do that.
helpful.
Okay. You know what? Give me a second, okay? Just give me a moment here. I used to do that, but then people don't really
Right. I'm old
utilize it.
and the sciences are very new for me, so
Yeah, no, that's fine. I mean,
Yeah.
like you guys can ask me too, right? Like you can. I mean, I'm only here for 1 more week, but then like feel free to email me. We can set up like one-on-one what do you call that? One-on-one like help, right? Or if you guys want to come for help as a group, we can do that. We can do it in person. We can do it virtually. That's not a problem.
thank
at
you,
all.
Vincent.
Yeah,
I appreciate it.
just give me a second, okay? I just need to set this up real quick, and then you guys will have a way to communicate. GS 182 2023 summer. Copy. Okay, I'm gonna put it in the chat box. All right, can you guys click on it and see if you're able to get there? Okay, so like I'll get a notification when you guys post on there. Like you
Yeah.
probably see my, yeah, it's blank right now, right? So like here, I'll bring it over here. So you see, right? I just posted that. So if you want to post something, you can click on the plus here, right? And then you type a subject, you know, like question about cellular respiration or something. You can share your notes. If you took really good notes and you want to share with the class, you can upload it, right? You can write your question here, and then we'll be able to see it, right? Yeah. So I already see some people are typing here, right? And then you can actually you can answer the post, right? Like you can yeah. So yeah, but don't just put hi, right? Like then I'll be getting like 20 emails saying, you know, someone say hello on the post, right? So you can answer each other's question, right? And that way, you know, it's an interactive situation, right? If you guys have like study groups, you know, you can feel free to organize it here if you have like a chat box or something.
I'm
chat
sorry,
group.
Vincent. How do I find this? My screen disappeared and I lost.
Oh, that's okay. I'll post this on eCentennial as well. But
Okay,
right now,
okay.
I just posted in the chat box, right?
Yeah.
Okay. Now, what I do not want you guys to do is like don't post like test question up here, right? Okay. Like if I see people post test question here, I'm gonna have to disable this thing. Okay. But you know, it give you guys a way to you know, to communicate. Okay, if you want to find each other. Hopefully that helps. All right. So let's go back to Krebs cycle. Where was I? Right. So the story so far, right? The story so far goes like this. And I know I'm repeating myself, but it doesn't hurt, right? So we have the glucose, which was 6 carbon. Then we get the pyruvate. And by the way, you guys get free tutoring services from school, right? You just have to book it at the library. Did anybody tell you guys that yet? The tutoring service?
Yeah, I try, but I get rescheduled every time.
Oh, okay. Well, that's out of my control, but you know, that option is there, should you need it, okay? For not just biology, but chemistry and math and you know, English as well. Okay, so we got our pyruvate, right? And then what happens to the pyruvate? Well, the pyruvate becomes the acetyl-CoA. What I recommend not to do is don't go on YouTube and search up this thing and watch it. Okay, I am trying to what's the word? Like I don't want to say dumb it down, okay, but like I try to take away like extra things that you don't know. And if you watch some YouTube video online, you are very likely to run into like more complicated information. Okay, that's gonna make it even more confusing for you. Okay? So just watch the recordings that I've given you. You know, go through the PowerPoint slides. And you know, if you're stuck, just email me. For those of you who have emailed me, you know I reply you like super fast. Okay, so do not hesitate. All right, so let's try to go back to this thing. We have the acetyl-CoA, which is 2 carbons, right? 2 carbons. Hopefully you know that by now. And we're still in the matrix, by the way. And for reasons that you don't have to really fully understand, there is a molecule that is in the matrix already. Yeah, that molecule has a name. It's called oxaloacetate. The name is not too important. What's important is this oxaloacetate has 4 carbon molecule atoms in it. Okay. So when the acetyl-CoA is formed, it will combine with the oxaloacetate. Combine with the oxaloacetate. So tell me, how many carbons are we gonna have when you take the acetyl-CoA and combine it with oxaloacetate?
6.
6, very simple math, right? 4 plus 2 gives you 6, and you get citric acid as the result. 6 carbon. That's why this is called the citric acid cycle, because the first product you form is citric acid. It's also called a Krebs cycle because the person who supposedly discovered it, his name is Krebs. Okay, so long story short, many steps are going to happen, which we don't have to memorize, fortunately for you. But the key things to know is that along the way, we will be, as we did in stage 1 and stage 2, releasing high energy electrons. Every time we release high energy electron, we are going to be capturing it, right? Okay, so we're going to be supplying NAD plus into the cycle. In fact, we're going to be supplying 3 of them. And we haven't seen the other energy carrier so far. That one is the FAD, right? Okay, so we're going to be having 1 of them. And once it goes through the cycle, on the other end, we're going to spit out 3 NADH and 1 FADH2. Okay. And then also, we will be producing ATP, 1 ATP. Right. So this over here, this is by substrate level phosphorylation. Okay. And at the end, when you have when you have broken down the citric acid cycle, we would also be releasing the remaining 2 carbon dioxide, right? The 2 carbon dioxide came from the original 2 carbon from the glucose, right? So, you know, you get the 6 carbon, but 4 was not from glucose, 4 of them was from this molecule. And once you remove the carbon dioxide, 6 minus 2 takes you back to 4. Okay, so then you regenerate oxaloacetate, which will be available to react with another acetyl-CoA, right? So remember, this whole thing happens 2 times for each glucose molecule that you metabolize, because each glucose molecule gives you 2 pyruvate, which gives you 2 acetyl-CoA, right? So I'm just going to copy it like that. You can reorganize the notes at a later time. Right? So like that. There we go. Okay. So let's write down the main products here. Main products. Right. So we have 6 NADH, 3 from each cycle, 2 FADH2, 4 carbon dioxide, and 2 ATP. Okay. Now I know you guys don't have a lot of head space for this right now because you have a quiz today. So focus on the quiz first. You know, and then when you're done the quiz, organize the notes again. And then on Wednesday, we will have a quick review of this material. We'll finish the lesson, and then hopefully you'll feel better about it. The question on the test regarding these concepts, they're not too difficult. Okay, they're fairly straightforward. Okay. So good luck with the quiz today. Make sure you do it before 11:59 p.m. And I'll see you all on Wednesday. Oh, there's a question. Yeah, go ahead.
Yeah, so can I ask you a question from
Yeah, sure.
Yeah, it's on okay, I'm sorry, osmosis, diffusion of water. Yeah. So on hypertonic, when the movement is hypertonic, so it says the concentration of solution is greater than the concentration of cytoplasm. So it's a concentration means is it a concentration of water or is it something?
When I say concentration of something, is always concentration of the solute. Okay. Yeah. So like in did you say hypertonic? Yeah, hypertonic. So that means there is more stuff on the outside, more solute. Okay, so these are like, you know, NaCl maybe. okay, compared to the inside. So in this case, if it's hypertonic, you will be losing water, right?
Yeah.
So the cell will shrink, right? We call that plasmolysis. All right.
When we compare the solution, how do we write it? Like, that's the point I get confused.